Ultrasound processing. Before performing the experiment, it is important to sterilize the opening of the jar using the flame from a Bunsen burner. Circle the flame around the mouth of the jar to eliminate bacteria in the air from entering the sample. The setup for ultrasound processing is relatively simple. Both the sample and a basin is placed on an adjustable stage directly under the sonotrode. Adjust the stage such that the sonotrode is immersed in the inoculated liquid. Remember to leave a small gap between the base of the sonotrode and the base of the jar. Fill the basin with ice and water to create an ice water bath. During ultrasound processing, great amounts of heat is created. The heat generated will kill the bacteria, hence the ice water bath is needed to cool down the sample to prevent elimination of bacteria. In this experiment, the aim is not to kill the bacteria but only to injure them and observe their growth after processing. Furthermore, to prevent any accidents from happening during the experiment, parafilm strips are used to bind the glass jar to the sonotrode to stabilize the jar. Once you've ensured that the experiment has been properly set up, press the start button on the program to initiate the experiment. Also, remember to close the door of the ultrasound machine and wear earplugs when ultrasound processing is ongoing. Ultrasound waves have very high frequency that will damage your ears. After processing, sterilize the sonotrode by wiping it with tissue paper soaked in ethanol. This is important as to avoid cross-contamination that may affect the accuracy of your results. Experiments involving microorganisms are usually performed in a laminar flow to prevent contamination from the surroundings that can lead to inaccurate results. In addition, it is always advisable to sterilize any equipment and gloves before placing it in the laminar flow. Prior to plating, a serial dilution is performed to dilute the samples such that it is within countable range to allow us to estimate the total population of bacteria that survive after processing. To perform a serial dilution, 1 ml of sample is added to 9 ml of peptone water to achieve a 1-factor dilution. 1 ml of diluted sample is further added into 9 ml of peptone water to achieve a 2-factor dilution. This procedure goes on until the required dilutions are achieved. Now that we are done with serial dilution, we can proceed to plating of the agar. There are two methods of plating, spread plate or pour plate. In this video, we will be demonstrating the technique for pour plating. Pour plating is usually used for lactic acid bacteria as it creates an anaerobic condition for the bacteria to produce lactic acid during anaerobic respiration. In pour plating, 1 ml of sample is transferred onto a petri dish before pouring a layer of warm agar over it. The petri dish is covered, and using your fingers, move the petri dish gently in a figure-8 motion clockwise and anti-clockwise to allow the sample to be evenly mixed with the agar. The agar is left to cool and solidify before a second layer is added. The petri dish is then placed in an incubator at 37 degrees Celsius for 48 hours before counting. To determine the number of bacteria present after processing, a plate counter is used to quantify the number of colonies present on the plate. The units of plate count are total colony forming units per plate, and this value is converted into colony forming units per milliliters or grams of sample, based on the equation learned in food microbiology courses. The plate counter is easy to use as all you need to do is to tap the petri dish with a marker and the count will be registered.